This is Brother Barnes, and I just want to welcome you to this true and false quiz game. Before we take the quiz, though, we need to learn some basics so that we can even understand what this quiz is about. I want to talk about CRISPR technology. It is the acronym for Clustered Regularly Interspaced Short Palindromic Repeats. Well, what does all this mean? Before we get to what it means, let's talk about some DNA rules just for a moment. Remember that when we base pair in DNA, the adenine bonds to the thymine, or vice versa, thymine to adenine, guanine bonds to cytosine, or vice versa, cytosine to guanine. Also remember that DNA runs in an anti-parallel configuration, 5' to 3', 3' to 5'. The strands are mirror images of one another, and they run in opposite directions. And the 5' to 3' just simply talks about the location of where the phosphates join onto the sugars in the backbone of the DNA. That's what this numbering 5' to 3', 3' to 5' even means. That's what that means. Let's get down to CRISPR, though, just for a moment. There were scientists back in the 80s that kept noticing that there were repeated portions of bacterial genomes that they didn't really understand. This was back in the 80s. Fast forward into the 2000s, what eventually was determined, or what was found out, was that the DNA that was between these sticky ends, this was actually a mugshot of some virus that they had encountered before. So it was like a memory system that these bacteria had. That leads us to the word palindromic. Well, what does palindromic mean? It means that if you force these segments together, this T matches with this A, this T matches with this A, and then if you flipped this segment around, instead of going 5' to 3' on the top, if we go to 3' to 5' and read it in the opposite direction, we still have a T matching an A and an A matching a T. We still have the same thing. So basically, the bacteria were, in a very clever way, placing these snapshots of the bad guys into their genome so that if they encountered this bad guy again, they could deal with it. What has happened, though, in the late 2000s and on into the teens, though, what has happened that is so amazing is that if this segment can be pulled out of between these sticky ends, then scientists have figured out that they can insert the gene of choice into this spot. Basically, a cut and paste mechanism. It is crazy, unbelievable, the potential that CRISPR technology has. So, with that in mind, let's don't keep going, because a 3-minute prep for a simple true-false question is quite a bit. Here is my true and false question. Is CRISPR the acronym for Clustered Regularly Interspaced Short Palindromic Repeats? Is that correct? Thumbs up for true, thumbs down for false. I will be leaving some helpful links in the description below the video so that you and I can learn more about this together. Keep coming back.